Namaste everyone. Namaste Giri sir. Very good evening everyone. I take immense pride, more than a pride, more than a kind of a honor, in having Professor Giri Polavarapu sir, who is a mentor, more than a kind of a philosopher and a guide, and a person behind many avenues, not just mere into genomics or diagnostics, but for that matter, he has been an epitome of success in whatever fields that he has kindly chosen. I'll just start with a brief introduction about Giri Polavarapu sir. He has taken his immense time. In fact, I've delayed this particular civilization for some good reasons, because those particular good reasons always come when our morale is down. Whenever we feel that our morale is down, it's always nice to talk with wonderful person like Giri sir. Giri sir obtained his PhD with prestigious Professor Nayudamma, who was an ex-DG CSAR way back in early 1980s, long before many of us were born, probably. And then he went on to join Penn State University, and that was where during Penn State University in mid 1980s, sir went on to develop a lot of rapid diagnostic kits, genomic assays. He worked on various multifaceted disciplines in the areas of cancer biology, diagnostics, and now what we call it as theranostics. And later on, he worked for CDC, Center for Disease Control, US, as a kind of an advisor. And he has been, continually serving as an advisor for CDC and NIH USA. And that was where in mid 1990s, he established this Genomics Biotech, where he shaped Genomics Biotech company, where he heralded many mentees, where at least 11 different kits were developed. Be it the lateral flow assay based diagnostic kits, the genomic assays, and several other things, where he mentored and established those kind of things in various spheres and avenues, largely in the areas of animal biotechnology and diagnostics. And amidst these kind of things, sir also served as a visiting professor at Osmania University, Vignan University, and now he has been a professor emeritus at MIT University, Rajasthan. And sir needs no special mention. He is a younger brother of Professor Kavikishore sir, who is also here. In fact, when we look at both of these wonderful personalities, we draw a lot of inspiration. They have tremendous respect, for each other. They have tremendous respect, for the science, for the community. And for younger people, for so-called amateurs like us, where they respect with someone like a prefix like doctor and Garu or G every time. I don't know how on earth or we can really ask for. That itself is a kind of a blessing, in the form of every time persons like Giri and Giri sir and Kavi sir speaks. So thank you so much, Giri sir, for joining us. We'll get started with this. Thank you. Thank you so much.
Thank you very much, Dr. Prash. Well, as you said, I started my career very early in late 70s. In 78, I started joining the PhD, and 82 I finished it. With Professor Nayudamma at CLRI, and then I moved to NIH for 9 years. I was at NIH, National Institutes of Health in Rockville, Maryland, Bethesda, and then I moved to Penn State, and from there to Emory University, Core Labs. After September 11th, I joined the CDC. After that, 5 years working with the self agents establishing all the bioterroristic agents, and I moved to MWG Biotech as a director of operations. And for a short time, I was also as MD, and then I started moving on my own. I took the very, I don't know what you call, a very brave step, and I started, and I didn't turn back. Now I am here, and we have three companies. One Genomics Molecular Diagnostics, Genomics Biotech in USA, and one is Genomics Carl. And we have three companies, and Carl is more Center for Advanced Research for Livestock. Molecular Diagnostics is human, most of it. Now you can ask me the questions, and for me, throughout my career, and I always take my brother, Professor Kishore, though we were together from siblings by kids. He used to take care of me, and he was senior to me in the same high school, same college. And then he moved into plant side, and I moved into animal side. And then we started, after some time, then we started working in the oxidative stress and stress tolerance, and a lot of common things. Then we started working together almost like 15 years together. We don't see much difference with each other, except he is more into the plants and I am more into the animal and human healthcare.
Wonderful, sir. Thank you so much, sir. We'll have a few questions. So the very first question I wanted to ask you is, sir, what transpired you to become a kind of an entrepreneur? Because you were at the helm of affairs, you were at CSIR, you were doing with a PhD with prestigious Professor Nayudamma. What was the raison d'etre, sir? At that point of time, probably no one would have really thought of moving towards an industry. It's certainly kind of a dared step at that point of time. What drew your inspirations for that?
From the PhD time itself, I always feel like I want to do the translational research rather than basic research. Though I am interested in the basic research, I end up doing a lot of basic research, and cloning and expression, purification of the proteins, and regulation pathways, and heavily I did for 10 years. But then always I look at product developments, and I tried several things when I was in the PhD time, including the wound healing patches, and skin caring cosmetics, because I am from CLRI. And also, I did a lot of things like anti-venom experiments, though my work was on insect cuticle organization and pesticide development. But I always tend towards the translational research. Then NIH life is very busy. 18 hours to 20 hours we work in the day and night, sleep in the lab. After coming to Penn State, I was a little bit relaxed. So at that time, suddenly the automated DNA sequencing started coming in early, late 80s. And we are the first people published sequencing, cloning and expression and sequencing without using any isotope. So that is the first time we used all the chemicals, only chemicals, and used the instrumentation to do the automated sequencing rather than though it is a Sanger method. But then we started the first time a core lab at Penn State. I established it. I started seeing charging the people and getting the money into the core lab and making it into self-sustaining. Then when I moved to Penn State, to Emory, and then they gave me bigger operation, like a huge facility, more people working for me, as well as my R&D on a side by side. There I started looking at the working with a lot of people who are coming from different wide range of fields, from HIV to malaria, infectious diseases to the cancer biology. So they come and they meet with me, and then we design the projects for each. And then I started understanding in depth of every project, but not expert in anything. So I started doing the uh, first time we started doing the Evimetric chip, and then we started making our own chips. So that gave me a lot of momentum, how to prepare the product, how to sell it, how to design a, uh, how to make the money for the company. So we sustained by developing it, though I'm more secured because I'm a full-time employee from the government, from the university. So the institute is paying my salaries, but at the same time, we are running the core labs. So that gave me a lot of expertise about the how to manage the products, how to manage the service, how to deal with the customer, how to do the things. So though still I am academician, but I started having a little bit chip of a uh, mm, kind of a business attitude. Then when I moved to CDC, completely academics, and also there I got a pretty good chance to develop the kits myself for the self-agents, anthrax, brucella, and smallpox, and a lot of Burkholderia, glanders. So for 5 years, we did only developing the kits, real-time PCR, establishing them, do the field service. After that, then I joined MWG. That gave me completely a different ball game. I am a director of operations. The first day, the MD came and he resigned. He said, "You are the right person here. You take care of it because you have experience." And he's a MBA guy. He doesn't know much about the science. So he gave me the red plate, which is completely companies running in negative. So 67 employees. I have to work with them day and out, shifts. I sleep in my office. I see per day 10,000 sequences and 10,000 oligos synthesized. So that gave me, and then I started understanding what is the cost of production, how to make a product, how to ship it, quality control, quality assurance, all those things started because I have to pay attention because to make the company from red to green. It took 1 year. It took a lot of work from me, but that time made me very, very comfortable because I made the company from red to green. Then I came here and to establish another MWG biotech in India, in Bangalore. I looked at the market. I thought there is some market for the sequencing at that time. That was in 2004, but I was wrong. But my friends and my brother also, my brother was not encouraging me to join a company, to start a company, but he said, it's up to you. But I took the risk. I started the company, and my brother was there behind me and other friends, but I spent more time. I thought in 3 months I can establish and go. No, 15 years it took for me so far. Now I'm up to deep, up to here. So now we have 150 products, and we also writing the grants. I have almost like 25 PhD students within 15 years finish their PhD in India, and another 20 PhD students are in the process in the tube. So I feel very happy to have the students. I'm not entrepreneur alone, but I'm more academician turned into entrepreneur and count the penny, how to make the penny, what is the product. So what happened is over the time, in the beginning, I saw a lot of failures and I started keep on putting money. I didn't have much and I couldn't sell anything because what we make, nobody wants it. What people want, I don't know. So I started exploring. I moved completely into IVD, in vitro diagnostics. All those myths in my mind. Oh, I will do the sequencing, I will do the microarrays, I will do this, that. Consultancy, no, nothing will work. I realized people want simple, inexpensive, easy to use it, rapid diagnostics or anything. You can do it in the village. That is my motto. Then I started focusing on that. Malaria. Then where the money comes. Then I wrote the grant with DBT. Bairag gave me luckily enough money to start with. Then we got one by one, one by one. Now we got almost like 8 grants. That is making my students to survive. Then I started making the products. We started selling. And then I became more focusing on that and students PhDs. And the student PhDs are nothing but only translational research. So their focus is only to make a product. In the meantime, we started learning about all the regulatory things. To get approval from the drug control, we have to do all these things, all the steps. So I got that experience from MWC. So I applied here. So now we have almost like 150 products in both the companies here and there. Then we started seeing a big niche in the animal biotechnology. Nobody else is there in the whole country. So we are only the people. So I started focusing only on the animal biotechnology, completely focusing on that. Now we are going to move into the vaccines for the animals because there is a lot of gap, lot of demand. So this is all what I learned over the time is you can do the business, not what you want. You can do the business what the people want. Then make sure you can give it very inexpensive, even if you make it costly, but make a good product. So that's how now I'm doing both academics as well as industry also. But if you ask me which one you prefer, I don't know. I cannot tell. I cannot live without one.
Yeah, no, excellent, sir. I mean, quite phenomenal, sir. I mean, I know about 40 students doing PhDs in translational research, or who have done so many, 25 of them already, finished in translational research is quite phenomenal. We all know, sir, you have a wonderful academic setting, and your entrepreneurial values are also greatly valued. But at some point of time, sir, having worked for these prestigious institutes and having led wonderful projects, did you encounter any disagreements, sir, with anyone else? I mean, on any scientific terms, and yet, you might have converted those disagreements in a kind of an agreeable way. This is because this is the commonmost question I usually ask in civilizations, because the young generation needs to take some lessons. They need to learn how to disagree in an agreeable way with a mentor. That kind of learning is probably missing in India. We never experienced that kind of thing. Can you highlight one example from your experience?
Can you put the question again? I missed the part.
So I'm really sorry. I wanted to know from you, sir, were there any instances where you tried to disagree in an agreeable way with any of your colleagues on any kind of scientific experiments or any kind of discussions?
Yeah, always. Most probably, several times I have encountered. One is a very simple example when the COVID came first time, and everybody's panic. People are scared, and on March 15, even March 15th, first time when they started the lockdown initiated, and we closed the labs, but we are still working. And by the time I started working on the COVID, and they said, "Sir, we cannot work on COVID. COVID is very dangerous. People are dying in China and everywhere, so we cannot work." My uh, very serious colleagues, also my very close family members also told me, "No, don't dare. You are always going to the risky water and you do all these kinds of things. Don't touch COVID." I said, "No, I will do it, but carefully, elegantly." But that turned me completely different. Last 1.5 years, we made 15 years. I could not do the difference. I did a lot of difference. And also, I first time they called me, the principal secretary of AP government called me, sir, we called America, we want to get some expertise to some expert person who can help us to establish these labs to screen the COVID, because we are having a lot of problem everywhere. Nobody is coming and nobody knows how to do it. And he called me and said, CDC people told us that you are one of the experts in the country here, and can you help us? And my people said, don't go there, and they won't have money, they won't give. I said, no, it is my country, I will do it. So I went to Kurnool, established there with my car. Then I went to Anantapur, Nellore, Ongole, and Adoni, and Vijayawada, Srikakulam, Vizag, established all these things, and then I drove back to my place. I thought within 48 hours, every place, all are working very good. Then they called me, sir, why don't you start your own company in our place, do the service for the country, state. We jumped in. And at that time, my people, they started coming, yes, I think we can do it, because we started seeing you doing it, and so we will do it. So we did almost like thousands and thousands of samples screened, and we were very happy for that. But in the beginning, I had a very bad feeling from means not encouragement, but very negative. And the same way, and also I did several projects we did, we failed, and I thought it will work, because one is for snake poison. There is a plant, and they use that juice either in the eyes or in the wound. And I went and told to my mentor, Professor Naidama, sir, in my village they are using this juice for the snake venom bites. He said, very good. And since we don't know anything, why don't you buy some mice and buy some venom and inject and see, and use the. So see how a mentor can support you and also encourage you. If I tell, today if somebody comes and tells me, why don't you focus your own job, what you are supposed to do, rather than thinking about unnecessary things. But he didn't do that. He gave me the money. He asked me to buy it. I used it. No, I couldn't save any mice. And I tried sublethal dose. No, they couldn't survive. So what I realized is it is all wrong. What the myth about the juice. There may be some real plants which are more useful into this kind of thing, because it is all lipases inhibitors. So if you have a lipase inhibitor, it may work. But in this case, whatever I have, it doesn't have any alkaloids. So I realized I'm wrong. I mean, so in the beginning, I got excited, but by the end of the time, after killing 50 mice, I realized I'm wrong. I feel sorry even today. But that is kind of experiments we have to do sometimes. Hello?
Yeah, yes, sir. Yes, sir. Yes. Yeah.
But we never fight with each other. What we will do is, as you said, we agree to agree several times, and we may not agree. Sometimes people, they may say, no, we will do this way. And this, but with my experience, I will tell them, because a wounded soldier can tell more stories how to save your back when you get hit by an enemy, because he got wounded and he knows how to escape. So I'm one of the wounded soldier. I know how many times I burn my hands when I do some experiments. So if somebody wants to do that, I will tell politely. If they say, no, I want to change it, I will do it. Okay, I will give the chance to them.
I mean, wonderful, sir. I mean, I completely agree. I mean, that's a kind of a fascinating journey. Sir, moving further, the kind of rapid diagnostic kits or the kind of livestock kits that you have developed is quite phenomenal. Probably no one has probably developed any of these particular molecular diagnostic assays or kits in India than that you established. What was going through your mind, sir, when we really look into the livestock management in India? Why is that we couldn't develop or deliver those particular goods until you explored those kind of things? What was that element that was missing?
Okay, there are two things. One, If I turn back and why nobody is doing this, the question, it has a very clear-cut answer, because this agriculture, especially the uh, having a farming with the animal husbandry in a small house, is a household thing. But it is one of the most important for all the Indian farmers. 80% of them, they have one or two animals in the house, or a lot of buffaloes, or else sheep and goat. Uh, but the problem is, they are poor people. They cannot buy a kit. They cannot buy it, and only they have to rely on the government subsidies. So there is no retail price, retail market. But now, why I'm going into murky water? I started thinking about it. I got 4 grants coming from the DBT. DBT is looking at, hey, this is a gap in the country. Our farmers need these kits. And why can't we support this kind of research? Let him do it and we will support it. Yes, they gave me 80 lakhs, 40 lakhs, 60 lakhs, 1 crore. And then we made the kits, and then we started sending it to the animal husbandry department, and also sometimes to the farmers. They feel very happy when we did it. If you want to identify whether your animal has the brucellosis or not, and you don't have no idea, even the doctor doesn't even know sometimes. So if we give a test, just like a pregnancy kit, and they do it on the spot, they will know clearly it is a brucellosis. Then what to do? That is another problem. So this is what we started: a Brucella, Leptospira, para TB, bovine TB, wild TB, canine TB, and parvovirus, and we have mastitis. There are almost like hundreds of diseases. These animals, which they don't have a voice, they cannot tell their problem, but we can understand foot and mouth disease. So we started making these kits. Number one is satisfaction. Yes, I made the kit, and this is going to be useful for the country. But at the same time, glanders kind of thing, there is export value. So I look into that also. And there is a poultry. It is a huge business. Shrimp industry, there is a huge business. 40 to 80,000 crores business per year. So I look at the numbers. Where is the business? How I can move into that area? How better I can make a kit than existing kit? And how we can change it? Like I was thinking about the way Dr. Renuka is thinking about the aptamer based. Yes, aptamer itself cannot be done in the field. If we couple it with the lateral flow, yes, it can be a good essay, because it's a very sensitive, specific, and you can make it. A layman
Wonderful.
can read it. So I started focusing on that and looking into the animal industry, animal biotechnology, animal healthcare, in those things. And so nobody is coming nearby us. So we are doing those kits right now. So now I have to find out where is my web, where to whom I can sell. Send all the state government and give the whole list of the products what we are doing it, and then they can go and they can buy it, and they can sell it through the source.
Yeah, wonderful, sir. So moving further, I just thought during the next 30 minutes, I thought of discussing about the two wonderful research avenues that you have taken over the last 2 years, largely on COVID-19. Sir, you have developed wonderful COVID-19 kits, and you have established wonderful research avenues in the areas of seroprevalence and other areas. So can we say at this point of time, sir, that COVID-19 is going to be endemic at some point of time from now? That is number one. And number two is, if this endemicity is there, would we still be able to see the kind of waning immunity during the next maybe 10 years? Whenever we keep on studying this seroprevalence. Thank you, sir.
Yeah. COVID-19, when it came from the Wuhan, in the first month of 2020, because that's where we saw it, though it came in 2019 in China. India hit with 20. First thing what we did was, that's where the what we did. We sat together in the lab, and Janardhan and me, and asked Janardhan, pull all the sequences of the COVID-19. He said, sir, I don't see any sequence. I saw only SAR-MERS. I said, okay, let us go and find out in NIH websites. And they suddenly put it, NLM, and we pulled all the sequences. Then he said what to do. I said, okay, first let us see what are the immunogenic epitopes. Then we took the spike protein, N protein, envelope protein. Then we looked at that. Each one is 650, 700 amino acids. It is too huge. So what we did was, we put a couple of heads together and we pulled all the immunogenic areas and synthesized the whole gene, around 35 kD protein, which is not real protein. It is a synthetic gene which has nothing but 100% homology with areas of the COVID-19, either spike protein, N protein, or envelope protein. Why did you think that way? We know spike protein, it is an RNA virus. Spike protein will undergo a lot of modifications and mutations. And so let us take internal protein as well as N protein as well as envelope protein, and let us put it together. And we put that one, it stands still as a gold standard for my lab to look at the whole scenario. That started, we started raising antibodies. Then we started seeing the first patients. Then we saw very good immune response, and we can detect it with our synthetic protein. And we started making bulk of it and started using that synthetic protein for serosurveillance, both ELISA and rapid. This happened in 2020 before Zoom. Everything is done. And then we started giving for free. We never used it. In the meantime, then you guys started coming into the picture. Both you started putting a lot of big think tank together. And then we started thinking about the COVID and spike protein binding site ACE2 receptor. And then we started thinking about the curcumin. Curcumin became one because Professor GP is also interested. I am interested because of the curcumin. I lived in the curcumin from birth, because in my house, my dad grew healthy all the time, and my brother and me, we go and sleep in the curcumin powder, the whatever we call in the fields, farm. So we know, and we know it is a wonderful compound, and the antiviral, antibacterial. And here we know it works. So then Dr. Jalaja and Anil, they joined, and they started making tons of tablets, and we gave it for free. A lot of places we gave for free. We never made money. But that was a positive sense we had it. And now, to answer your second question also, that gave the momentum. Then I went and made the kits, and now we are also making the influenza A and B. The reason is, first wave, second wave, third wave is very, very slant, because I don't see much mortality, only morbidity, heavy morbidity, only 2 days, 3 days. But everybody is highly, either they are herd immunity is there, people already got it second time, third time. So it became like a normal flu in the society. So now the pandemic is over, gone, no more. Now it is all going to be endemic. Now what controls the endemic terms and those things we have to see next year. Whether it is the seasonal or regional, why Kerala is having more and why Maharashtra has more, is that is the biobehavior or the weather, we have to see. The second thing is how long this is going to be there. How is our immunity system? I think we have to think about other SARS and MERS scenario. And we, luckily, the world, WHO or FDA, CDC, all these people, they put their hands together. they were very loose in giving the vaccines licenses for emergency purpose. That also turned a lot of things better. Otherwise, we should have faced a lot of problems. Now, I don't think this will become more dangerous, but we cannot tell. And even if it comes, it will be sporadic. The other thing is, yes, this will be there for another 10 years. It may disappear like a JICA. And the other thing is, just like influenza A and B and pneumonia and this thing will be another SARS and MERS kind of thing, which is a ARDS, acute respiratory distress syndrome, will be in the vicinity, in the world, running around. So that is my feeling. I cannot tell. But immunity, I don't know. I have taken 5 times vaccination and I never got any symptoms so far in my last 3 years. I never got sick. I don't have it. Then if you look at my antibodies, I always have IgM, IgG. It means I might have infected, but I cannot show any symptoms. I never cough. I never had any sneezing, runny nose related to that. So it is very strange. So every 6 months you take another booster, most probably we will be fine.
On a lighter note, sir, the virus would definitely fear a lot in coming very closer to you, because you have been that particular enigma behind development of several kits. It's not an easy thing to attack Professor Giri Polavarapu, sir. Yeah, that's quite phenomenal, sir. That's quite amazing, sir. Something on these particular lines. I mean, as soon as this particular virus came into picture, I know you and Professor Kavikishore sir and Professor GP sir, you developed wonderful curcumin tablets, and you call them as GP kits, and all those kind of things. And you were also the very first one who developed these curcumin masks as well, which is quite phenomenal. I mean, if you really see, these are all, I would say, uh, uh, very big challenges look very easy, sir. I would say, yeah, exactly. So friends, if you really look, that is what sir is showing. Can you ever think of curcumin mass? Where you really want to ensure that the viral load in your oropharyngeal cavity or nasopharyngeal cavity should be abrupted. That's where sir has developed those particular kits in a matter of no time. That is very fast. So can you tell us how or what inspired you to develop those things very fastly, sir? Thank you so much.
Okay. Curcumin is, we know very well, and you see the problem with curcumin is there are it is a two-edged knife. A lot of people, they hate curcumin because they think like, hey, we eat it every day. Why do you make it as a medicine? It doesn't do anything. It is only food. To our food coloring agent. But some people, they look at the structure of the curcumin and they look at the dynamics of the curcumin molecule and what it does. Antiviral, antibacterial, antifungal, and also it has a lot of other things, including anti-malarial activity also there, if along with other things. And a lot of people, malaria is sometimes fatal, especially in cerebral malaria, and also pregnancy issues with the malaria. And those pregnant ladies, they have malaria, means they are really fatal for the baby. So curcumin helps. And it is not like me believing it, it is also the fact. And if you believe in the facts, then you stand for it. The same thing with the GP. Professor GP is Padma Bhushan, and he has been working his whole life, 40, 50 years, he worked only on the curcumin based things. And he and me, we sit together on the phone for hours together. We discuss about these things, and one by one, the tablets, first thing we did. And he was really surprised. And then I said, okay, why don't we make the mask? He said, that is a great idea. Then I put the GP mask. Then bags in Tamil Nadu, now they are giving for free bags, curcumin bags, yellow bags, for free in all over the Tamil Nadu state. And then now curcumin goggle, and they are putting in the mouthwash. and they are using that. So these are the products which spontaneously we think, and we see whether it is possible to make it, and then we go after it. And it is not for the people. And one day people may ask it. Now in America they started asking for the Kirkman gloves, Kirkman mask, and Kirkman oral fluids, uh, because they are scared of COVID, and they think Kirkman will work, because a lot of this what we call is organic stores, they are really selling Kirkman like crazy, the tablets and powder and everything. So everybody is buying in America and Europe also.
Wonderful, sir. Sir, something on these COVID-19 again. Sir, when alpha, beta, epsilon, and even delta variants could be detected using RT-PCR probes, for some reasons, we couldn't muster the support, or we couldn't develop those sophisticated probes for Omicron variant. We missed that particular bus. So let's say, Shom, now yesterday we had Delta Cron, which is a new variant that has come up in US and in Europe. And say 5 or 6 years down the line, we might be getting, say, another 10 such variants, maybe Zeta variant or ZTF variant. Can we at this point of time predict any such kind of probes for these particular variants? And maybe if not the RT-PCR, can we be able to develop some kind of home-based lateral flow assays or rapid antigen tests, which could be detectable even if some new variants that pop up in the near future?
Yeah, it is a twofold. The answer is in twofold. Number one, and we are also funded by DBT last year. It is a past phase funding by Jigsaw and Genomics. And what we did was, the Jigsaw people are 100% only bioinformatic people. They don't have any wet lab. What they do is they sit in front of the huge computers and they look at all the database and coming in thousands and thousands of sequences, and they compare with that, and they come up with whole genome of the COVID. And they came up with a small area, which is 100%, 99.9% homology with all of them, highly conserved area. And we used that one and made the real-time PCR probes, primers. And that is working very well. So that will recognize 100% COVID only, including alpha, beta, delta, gamma, epsilon, Omicron, all of them. That is one part. And we submitted to ICMR, and now they are supposed to ship it to NIV to validate. That is the one part. The second one is, as I said before, and the one which we developed the area of different areas, not one, because this is a very strange bug, and it undergoes a lot of modifications. And even 5 or 10 nucleotides are variant from here and there, it changes a lot of amino acid structure. And that gives you very false positive or false negative, most of the time, false negative. So what we did was, we took different areas which are highly conserved, and then we also took randomly select zero surveillance based areas and put them together. Now we have two different clones, what we synthesized. One is having the spike protein, envelope protein, and E protein. And now we started making small synthetic peptides, only Omicron and other things also, adding that into this and raising the antibodies in the chicken, using those antibodies to detect the Omicron. So far, we didn't miss any of them. I will ship you, if you want, I can send you a bunch of boxes. And right now, I never heard anybody missing one COVID case by lateral flow. But that there is a catch. Which day we have to pick up? Maybe after infection, second or third day. When they have the cough, that is the best place. And you can take the nasal or oral.
Yeah, wonderful, sir. So personally, I feel, I mean, since my specialization is next generation sequencing and systems genomics, after having analyzed some X number of genomes, even the SARS-CoV-2 genomes, I think this next generation sequencing based approaches are becoming very farce, very wrong, I would say. I mean, because even though we are able to detect that SONS is a kind of an Omicron variant, we are not able to move further beyond that kind of sequencing regimen for that particular variant. So, for example, and at the same time, many countries are investing huge monies towards these sequencing facilities. And for example, even in India, we are thinking of having a kind of a sequencing center every 500 kilometers, which almost, in many states, we have already established this. I don't know if this is really going to be a bit hype, or are we really taking this kind of approach for granted, or are we really going to bring up some kind of means for a better diagnostic surveillance in the future, sir? What do you think?
Okay, NGS is a shooting in the dark kind of thing. That's what you are saying is the false thing. Now, there is another way of doing it. Let us go back original, our true gold standard Sanger method, using the applied biosystem probe, and so where we have all the 4 nucleotides are labeled with 4 different dyes, rock tamra and other stuff, and use the same thing, but use the specific primers. You need only, for Omicron, you need only one primer, which can go through this Omicron variant region that can tell in a one single sequence. You don't have to do. You will do RNA extraction normal, reverse transcribe is normal, then you do the sequencing by using the specific primer, either forward or reverse. You can 100% pick up Omicron variant. You can't miss it. You don't have to put too much money. It costs only maximum. Nowadays, it is only $5 to $10 per sequencing reaction. So I think even last week I was thinking seriously, we should come up with a set of primers, do the only Sanger method, and do the sequencing just like by using the Sanger method technology, by using the capillary sequencing rather than NGS. NGS takes, means we don't have too many NGS, and there may be NGS, but to do that, it may not be picking up all the things.
Right, wonderful, sir. I mean, I have this minion on my table. This is a long read sequencing technology. We use this, and yet, we are still not seeing that kind of success rate, despite the fact that the long read sequencing like minion also does some kind of a lot of erroneous rates. We are still not really reaching that kind of success rate with this, unfortunately, with NGS. And I think we are spending a lot of money. We are not really reaching that kind of pinnacle of success. Yeah. Thank you, sir. Sir, the next 10 minutes, I thought we'll have some question and answers from the audience as well. But first, to draw more inspiration, I thought Professor Kavikishore, sir, if he has any questions to his younger brother, to his wonderful person in the form of you. Kavikishore said, do you have any question for Giri, sir? Yeah, we have 2 wonderful personalities.
Yeah, I don't have any questions, sir. But one thing I should tell you about Dr. Giri, he's always courageous from childhood onwards. And not only courageous, but also confident about what he is doing, which I lack. Certainly, I lack that. And then, I must also tell you that, he dared to go to Professor Naiduma, a personality, a towering personality, and approached him, for his PhD. And Naiduma has given only, I think, 15 seconds or so. He was, going on the steps, and he wanted him, to talk to him, what he wanted to say. And Dr. Giri was given only hardly 13 or 15 seconds, not much. He informed him everything, and then Professor Naidamma said, okay, I'll give you 45 minutes or so. You go to the library, refer to the library, and get back. and come with some solid objectives and also the hypothesis, which Professor Giri did it successfully. I could not have done that. That is what Dr. Giri is. Same thing happened to me recently. I went to IIT Hyderabad, and the director of IIT Hyderabad did not give me any time. I was sitting there for 3 hours nearly outside his chambers. And two PAs told me that it is impossible for you to meet him, because he was always busy with several meetings, one after another. Then finally, one PA came to me and then said, sir, he is going out for another meeting, and you'd get only 15 seconds or so to talk to him. By the time, he leaves the chambers and go to his car, you would get some 15, 20 seconds. You talk to him whatever you want. After talking to me, for 15 or 20 seconds, he said, no, 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 you get into the car. I want to talk to you more. And he took me to his house also on the same day. We had a lunch together and all that. That is how, things, happen. So that's what I wanted to say. He is a very, very courageous guy, Dr. Giri, and very confident guy, which I lack. Thank you so much for giving me an opportunity.
No, at all, sir. No, at all, sir. I mean, that speaks a wonderful, impeccable respect that you have for each other, sir. That's wonderful. Thank you so much, sir. On a lighter note, probably what Giri sir achieved some 45 years ago when he was doing his PhD with Professor Naidama, probably you could do it now, maybe 2 months ago, at the helm of maybe 73 years or 74 years. That speaks volumes about Giri sir. Thank you, Giri sir. Thank you, Kavi sir. Yeah. Indrani, do you have any quick question, please? Please be very quick. Sir has another
Yes,
meeting. Yeah.
yes, sir. It was such inspiring session from Giri sir. I had two questions. So, sir, since you're working on the translational side, so what all types of in vitro diagnostic kits that you have designed still now for cancer patients?
Okay. Okay. Now there are Indrani. I think that is Miss Indrani Biswas.
Yes, sir. Yes, sir. Yes, sir.
Yes, I remember you are working on
Animal models, sir.
immunosuppressive. Yes, animal
Yes,
model, and you
yes,
are
yes.
having
Exactly, sir.
yes, I told you to use the cyclosporin to immunosuppress because it
Suppress
is
the
one of the,
animals.
yes, and it will suppress the calcineurin, and calcineurin is the which is the modulator for the immune response, and that will block through the cyclosporin. Did you try that experiment? I just want to.
Yes, I'm just working on that part, sir. Parallelly, we are planning to get one more set of actually animals for new experiments. So I'm working on that actually now.
Yeah, because the nude mice are nude. These things are very expensive, and also you need a very clean room to do that kind of
Exactly,
thing.
sir. Yes,
And
sir.
we
Yes,
did
sir.
that experiments, but here in this country, if you want to do it, you have to think as an Indian, not
Yes,
in
sir.
America.
Exactly.
Yeah,
Exactly.
so these things, this may work. The other thing is
Sure,
you
sir. Sure.
asked me about the cancer markers. Yes, there are almost like including the breast cancer markers, what we call is new and hair too, and we have PSA, prostate secretory antigen, and also we have several, almost like uh, I think we have almost 28 different cancer markers we are making right now. Lateral flow kits, as well as we are also doing the ELISA
Okay, sir.
markers.
Mostly you're working on the base of ELISA. I mean, as a fast diagnostic kits.
Rapid, both of them.
Rapid. Okay,
Yes.
sir. And my second question was like, since like seroprevalence was working as an most important topic in most of the respiratory diseases. So, how far do you think, sir, it can be applicable for cancer as well? To in the case of like, designing some biomarkers from prognosis or diagnosis point of view, sir.
Okay. Now that is an important question because some of the uh, these cancers are not that invasive. They are not invasive. So some are the
Yes, sir.
easy to diagnose, some are not easy to diagnose. And to get the body fluid is not that easy. But only people who are undergoing any surgery and the doctor may suspect, yes, this may be benign, this may be carcinogenic, cancerous. So they have to go and find out. So when they bring this biopsy, and that tissue you can use it to make your kit, your this thing. And also some of them, some cancers, when they become like metastasis, means they spread out,
Yes, sir.
then they may be coming into the bloodstream, and you can see at the picogram levels also some of the the epitopes which are not present in the normal cells, they are present only in the carcinous cells. You can find them in the ELISA kind of assays. The other one is once the tissue sample comes, and you can also do the PCR based assays also for these all the markers. This is well studied, a lot of markers, and there are a lot of diagnostic assays, both invasive, non-invasive methods.
Yes, sir. Yes, sir. Yes, sir. Thank you, sir. Thank you so much.
Thank
Thank
you.
you,
Yeah.
sir.
Sir. Thank you,
Thank
Giri
you,
sir. Anil, please.
Yeah. Thank you, Prashant, for giving this opportunity. I'm very lucky to work with both the doyens of the field, plant genetics and animal genetics, Professor Kavikish sir and Giri sir. Giri sir, when one small question. Being a successful academician, you move to translation research. If God gives you 30 years back of the previous 30 years back to you, will you stay in the same field, translation, or will you move to any other field?
This I have in my mind when I was early 20s, and I want to do aggressive. As my brother said, uh, he calls me courageous. For me, I'm a stubborn, and I failed, and don't take it. I am successful guy. No, I failed several times in the beginning when I came in 2005 and 6 until 2009. I was a failure. Failure in the sense I couldn't do anything much, and there is no help. I don't know what to do, where to go, and how to get the raw materials, who are the vendors, how to sell my product, and how to get the validation. I don't even know how to get the drug license, and whom should I contact, what is the bribing, whom should I bribe, should I bribe it or not, can I avoid all these things. There were a lot of questions are there. I can answer all those things for you guys when you talk. Yes, and from the beginning, I want to do it. I was a little bit more stubborn and also focused in the sense I want to do it. I want to do the business. I want to start. I want to make the product. That is the intention I had. Well, if I couldn't do it in 90s, and I tried in 2004 because at that time I am in my peak, 50 years old, and 2000s, and I was very aggressive, and I thought I want to take a risk. So 4 years I struggled. Then I'm okay. Then now I feel very confident. Now I want to move other things. What I want to do, like vaccinology. I want to start vaccines too because I got a little bit of momentum. Yeah, COVID really gave me a lot of momentum. COVID really helped me in changing my personality, changing my thoughts, changing my course, and also made me a better man in thinking.
Yeah, thank you, sir. It's certainly a kind of blessing in disguise. Thank you so much, Anil, for asking that wonderful question. Sugu, a quick question, please. Yeah.
Yeah, good evening, sir. I'm fortunate, I'm part of this sciversation with you, sir. First of all, thank you, Prash. And here I have my mentor, Professor Kavikishwar, sir, from my MSc. Sir, I have a question to you. You are an academician, you are an entrepreneur. So we are academicians. We wanted to become an entrepreneur like you, make a place a kind of difference for a kind of diagnostics in India. How do you feel like where we have to start? Budding entrepreneurs like me or Anil or someone like here who wanted to think of starting career as entrepreneur along with being academician? Please, sir.
Okay, now first of all, you sit yourself and think exactly, draw it on the paper, what exactly you want to do. Then do due diligence, what we call, means if I am making this product, how many people will buy? What is the market value? Is there anything or not? If you feel like, yes, there is a market, who are my competitors? Who are selling for what price? What is my cost of production if I make it? Okay. Making one product or making 10 different products, that is a different question. But if you are, I know about you and Anil, you are in the mid 30s, most probably, or else early 30s. And because you are a beginner, if you are moving out of the academics, that's okay, you can do it. If you have the confidence and also all these things, there is a small thing I can give you, my focus. First, number one, do that. Number two, don't leave the academics right away. What you have to do is go and write a big grant. Big grant in BIRAC will facilitate you with money to start a company. Hello? You understand?
Yes, sir. I'm listening, sir. Yeah, please, sir.
BIT, big grant, and Dr. Visu, he can help you. Vishwanath, he is in IKP Park, I think. Now he can help you to put the things together, and it may take 1 or 2 shots, but if you put a good proposal, you may get it. If you get it, focus on it, along with your academics. Then side by side, you start making it, put your extra time in the evening. Once you feel like, yes, I'm doing good, I can do, then you run in. Forget about academics. But imagine, once you go into entrepreneurship, you cannot come back into academics that easily. Very few people can do it, because the way I did it is, I finished my major academic run, and I am at the prime of my academics. I am already a full-time professor at Emory. I was already had enough papers, enough students are done. I don't have to satisfy my academic interest at that time. But at the same time, by coming into entrepreneurship as an industry, I didn't leave my academics. I took all the students again. I'm continuing that to satisfy my academic ego. You understand that? So I may be cheating by being in an entrepreneurship, but I did not really 100% entrepreneur. I'm still academician even today. So it is up to you, because in America we have the luxury of doing that, but in India I don't know whether you can do it. But there are people who are doing it.
Yeah, there are few universities in like ISIS who are encouraging such kind of things, sir. Thank you so much. Thank you, Sugu, for asking wonderful question. Ashok, quick question, please.
Yeah, thank you very much. I am really enjoying each and every step, the journey you are sharing yourself, being a journey of entrepreneurship. So I want to ask, we are working in this Chandigarh in Punjab University. So we have one drug repurposing molecule, and that was testified in silico and then in vitro studies, and tested on nude mouse model. And this particular drug is a very safe drug. Now we are struggling with some of the things, and it need to be go to the clinical setup. So as a person, in that sense, our university is not supporting in that sense. So from which step or from which direction we have to start this thing?
Okay.
As an academician, we get a lot of appreciation, but I want to convert into a low cost drug. In comparison to the present status, this particular 1 gram, this particular drug cost more than 50 rupees only, and it is giving more than 70% regression in this time, tumor suppression and regression. All these things are already been tested in my lab.
Okay, let me ask you this question: whether this compound is a Ayurvedic protein product, or it is allopathic?
So this is a fenbendazole, and this is a already known product, which is a deworming drug.
Okay.
Another product, yes.
Okay.
But it's
This
not been tested for anti-cancer activities.
okay. Now it is already established for the deworming.
Yes.
So it is a helminthes and
Yes,
helminthes
yes,
as a deworming for that to do
yes.
that. And we know how it works, like either arsenic compounds or any other normal compounds also do this sometimes. And also some of these compounds, deworming compounds, they work, but they also may have some of the anti-tumor activity also, just like
Yes.
curcumin. And curcumin also has some anti-tumor activity, but it is very difficult to prove which analog of the curcumin is really acting, because the whole crude curcumin, what we call haldi, it may be
Yes.
working, but we don't know which part of it. Here, I don't know which one you are dealing with. And
Okay.
if it is pure compound, then already established compound, so it is easy for you to go for the clinical trials.
Okay. So
And
we'll discuss in detail, sir. Thank you. I got this idea.
Okay.
This will
All right.
require a lot of discussion. Thank you very
Yes,
much.
yes.
Thank
All
you
right.
very much.
You
Thank
can
you.
call me later. Yeah.
Yes, sir. Yes. Thank you.
Thank you, sir. Thank you so much. Before we say one final goodbye, maybe Visu has a quick comment. Visu. Yeah, I'm sorry, Visu just left. But sir, thank you so much for your wonderful time and energy. I can only request all the members to switch on their video and
Prash,
have.
Very sorry,
Prash,
Prash.
Vishu is
I
here.
just got signed out, and very happy. No comments from my side, and we all are very privileged, sir. Like very few people in the academic background with the background and these kind of things they are doing. Yes, with the BAC grant kind of thing, I will be very happy to help people. But what you have done is very phenomenal. That needs a lot of commitment, and we will be very much inspired to and always looking at you, sir. Some of us always talk about you, may not be coming to you, but thank you very much, sir.
Thank you. All right. Thank you.
Thank you very much, sir. Thank you, Visu. All of you, please switch on your camera and have a wonderful photograph with both Professor Kavikishore sir and Professor Giri sir.
Thank you.
And have a big smile, say cheese, because it's going to be a rare photograph and a wonderful cherished photograph and a wonderful thing.
Thank you.
Thank you. Thank you, sir.
We have the third generation kits as well in the form of Anil and Giri sir just left. I don't know why. 1 minute. Maybe sir will join again. 1 minute.
Sure, she must be a young scientist.
Yeah.
Prashant sir has a meeting at 8:15. Probably he has left.
No, he has a meeting, but I think he'll be back. I think he lost his connection. I don't know.
Prash, if Sir is not able to join, please don't worry. I got a one copy. Yeah, Prash. No, you're in mute mode. You're in mute mode.
Oh, I'm sorry. I thought I thought I'll say some final words, and I thought we all should give a standing ovation to sir.
Exactly.
That was what I thought. And sir, lost the connection. So I think sir will join 1 minute. I think hopefully, otherwise I'll just give him a call. He said he has a meeting at 8:15. Yeah.
Good evening, sir.
Good evening, Dr. Sandeep. How are you?
I'm fine, sir. How is your health now, sir?
Yeah, better. You're at Bangalore only?
Yes, sir.
You're taking care of Dr. Jawahar?
Yes, sir.
Okay.
Good evening, sir.
Yeah, good evening, Dr. Altaf. How are you?
I'm good, sir. How are you, sir?
Yeah, I'm better now.
Master.
Good evening, sir. How are you, sir?
I am fine, Dr. Renuka.
It's
Sir,
really
you
inspirational talk
sir,
today.
you say
Yeah.
you are Usmania. You
Pardon
see,
me?
you are Usmania.
Good evening, Prash.
Okay.
Good, good evening. I'm 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 happy connecting. I'm happy connecting from Nigeria. So happy connecting from Nigeria.
Okay.
Love team. I'm very happy with you.
Thank you so much, sir. Thank you so much.
Thank you, Dr. Prashant, for organizing this one.
Thank you, Prash.
Thank you, Prash.
Thank you, sir.
Thank you,
Thank you.
sir.
Thank you so much, sir. Thank you, sir.
Thank you, everyone. Thank you,
Thank
sir.
you so much, sir.
Thank you, sir. Thank you, sir.
Thank you, sir. Thank you, sir.
Sir, you're in mute mode, sir. Yes, sir. Yes, sir. You're in mute mode, sir.
Okay, thank you, Dr. Prashant.
Sir, Giri, sir, you're in mute mode, sir. Prash, sir is saying something. Sir, you're in mute mode.
Maybe it's 8:14, so I think sir will be ready for the next one. So let us close this one.
Okay.
Thank you, sir.
Thank you, sir.